Telomeres, the protective caps at the ends of our chromosomes, play a central role in the biology of aging. Much like the plastic tips on shoelaces that prevent fraying, telomeres guard our genetic material from degradation and unintended recombination. With every cell division, however, telomeres gradually shorten, a process that eventually triggers cellular senescence, a state in which cells stop dividing and begin secreting inflammatory factors that contribute to tissue aging. This progressive erosion of telomere length is one of the most consistent biomarkers of biological aging, and it may hold critical clues to understanding both lifespan and health span. Research into the molecular machinery that maintains telomeres, particularly the genes HTRT, human telomerase reverse transcriptase, and TRC, telomerase RNA component, has illuminated how some cells and some individuals manage to slow this molecular countdown. Telomeres consist of repetitive DNA sequences, tag in humans, bound by specialized proteins that form a protective complex known as shelterin. Each time a cell divides, its DNA replication machinery cannot fully replicate the ends of linear chromosomes, resulting in the gradual loss of telomeric DNA. When telomeres reach a critically short length, cells enter senescence or undergo apoptosis, contributing to tissue dysfunction and the decline of regenerative capacity that characterizes aging. This process is counteracted, in part, by telomerase, a ribonucleoprotein enzyme that can add telomeric repeats to chromosome ends, thereby extending their length. Telomerase is composed of two essential components, TRC, which provides the RNA template, and HTRT, the catalytic subunit that synthesizes new DNA. In most somatic cells, telomerase is either inactive or expressed at very low levels, leading to steady telomere attrition over the life course. In contrast, telomerase remains active in stem cells, germ cells, and certain immune cells, helping maintain their proliferative potential over time. In centenarians and long-lived individuals, studies have found that telomere length tends to be longer than average for their age. Even more compelling, their hematopoietic stem cells, the source of blood and immune cells, appear to preserve telomere integrity more effectively, supporting continued immune function in very late life. Some of this preservation may be genetic. Variants in TRC and HTRT have been associated with longer telomeres and increased longevity across different populations. These genes influence the activity and stability of telomerase, and their expression levels can affect how quickly telomeres shorten in response to stress and replication demands. However, genetics is not the only determinant of telomere dynamics. Lifestyle and environmental factors exert a profound influence on telomere length. Chronic psychological stress, poor diet, physical inactivity, smoking, and obesity have all been linked to accelerated telomere shortening. Conversely, protective behaviors such as regular exercise, a Mediterranean-style diet, quality sleep, and strong social connections are associated with longer telomeres and slower biological aging. Interventions such as mindfulness-based stress reduction have even been shown to influence telomere maintenance, possibly by reducing oxidative and inflammatory stress that otherwise damages telomeric DNA. Oxidative stress is one of the main drivers of telomere attrition. Because telomeric DNA is rich in guanine, a base particularly susceptible to oxidative damage, telomeres are especially vulnerable to free radicals. This damage can accelerate shortening even in non-dividing cells, linking environmental exposures and metabolic health directly to genomic aging. Long-lived individuals often exhibit superior antioxidant defenses, which may contribute to telomere stability over time. Telomere biology also intersects with immune aging. As immune cells divide in response to infections or inflammation, they consume telomeric length at a faster rate. In older adults, short telomeres in T cells are associated with diminished immune responses and chronic inflammation, features of immunosenescence and inflammaging. In centenarians, however, immune cells often retain longer telomeres and more robust telomerase activity, enabling continued adaptive responses even in the 10th decade of life. Therapeutic strategies aimed at modulating telomere biology are now an area of active exploration. While indiscriminate activation of telomerase carries risks, especially since many cancers exploit telomerase to achieve uncontrolled growth, selective and transient upregulation in specific cell types, such as stem cells or immune cells, may offer ways to rejuvenate tissue without increasing malignancy risk. Experimental compounds Gene therapies and lifestyle-based interventions targeting telomere maintenance are being studied for their potential to extend health span and delay the onset of age-related diseases. Ultimately, telomeres are not clocks in the rigid sense, but barometers of cellular history and stress exposure. They reflect both intrinsic biological processes and the cumulative impact of life's choices and challenges. The fact that telomere length is modifiable and that it aligns so closely with functional aging offers a compelling framework for approaching longevity. In individuals who reach extreme old age with preserved vitality, telomeres seem to tell a story not just of time past, but of time well managed. Understanding and nurturing these genomic sentinels may be one of the most promising paths toward longer, healthier lives.